Okay, thank you very much. I too would like to echo the sentiments of the previous speakers in terms of indicating that we are very appreciative for the invitation to speak here today, and it's been great to meet everyone. And so I am one of the individuals that has recently come into the autism field also, and so I call myself, or rather we would say that we too have fallen under the influence of the Levitt trap. And but he assures me that when he saw some of our data related to animal models, looking at prenatal insult and postnatal dysregulation, that he was sure that we in fact could contribute to sort of understanding the molecular level mechanisms that may be operative in ASD. Right, and so I'd like to just tell you that story today. Start out with a little background, rationale, and then tell you about our hypothesis and approach, and then give you some correlates in terms of behavioral correlates, neurological dysfunction correlates, and then tell you about our MET work, and end up with a model and some conclusions. And so this then will move back and forth through this continuum of development, moving back and forth between animals and humans. And so I work with the compound called Benzopyrene. We call it BAP. And it's a quite common environmental contaminant. It is, as you can see here indicated on this slide, released primarily as a byproduct of combustion processes. From smokestacks in the industrial emissions. It's released from automobile exhaust. As you can see here, charbroiling, you are exposed to polycyclic aromatic hydrocarbons, okay? Of which BAP is the principal member of that family. Cigarette smoke, of course, has it, and municipal waste incineration processes also generate polycyclic aromatic hydrocarbons. And if your wife has you go to Hawaii during off season and you stay on the wrong island, you will also get exposed to benzopyrene from volcanic eruptions. Whoops. And that's the chemical structure of the compound there. And so it is an aryl hydrocarbon receptor agonist. The primary routes of exposure are oral and inhalation, and it has been shown to cause peripheral neuropathies as well as neurobehavioral deficits. And I'd like to tell you, start out today by telling you an epidemiological correlate here with regard to polycyclic aromatic hydrocarbons causing a decline in the mental development index in a prospective cohort in Harlem associated with Ricky Pierre's group at Columbia. And we'll just get right to it there. And so here, this was reported recently in EHP, and what Ricky did was continued one of the cohorts that has been ongoing since the collapse of the World Trade Center. And from this particular cohort, she put backpacks on mothers who were of certain demographics in Harlem. They had to wear these backpacks, monitors, if you will, from late first trimester to early second trimester, okay? She then followed these individuals as well as their children. And so what you see here is a plot of their mental development index as assessed by the Bayley Scales of Infant Development as a function of age. And so at 12, 24, and 36 months, she separated the groups into high and low exposure PAH groups, and found that at 36 months, she was able to discern a 5-point differential with regard to their ability to function on the Bayley Scales of Infant Development. Now, a 5-point drop in IQ points potentially represents a significant dilemma, especially if you extrapolate that through a particular population. And perhaps this is the type of cohort that Jeff was talking about, Peter, I'm sorry, was talking about this morning in terms of us probably needing to follow this cohort for ASD diagnosis also. And so this data, in fact, for the first time, represented a prospective cohort study, hypothesis-driven, if you will, Chris, that demonstrated that in utero exposure to PAHs affects early cognitive development. And so, of course, we were very happy to see this because over the last 5 years, the last grant cycle, our overarching hypothesis in the laboratory is that, in fact, prenatal exposure to these environmental contaminants actually produces behavioral learning and memory deficits. And of course, we think that those are mediated via glutamatergic receptor subunit dysregulation. And this, of course, we believe occurs when synapses are developing in early postnatal development for the first time, in other words. And so this is our approach. We call it, and have coined it, our susceptibility exposure paradigm. And this is our variation on the Barker hypothesis, which states that prenatal exposure leads to postnatal dysregulation of physiological processes. And it is based primarily on the fact that there's a critical window of exposure, if you will, for developmental insult, particularly with regard to peak periods of neuronal proliferation in certain specific brain structures like the cerebral cortex, the hippocampus. And so, as you can see here, entorhinal cortex, dentate gyrus is actually undergoing peak periods of neuronal proliferation from E14 to E17. So we devised a state-of-the-art inhalation exposure system where we generate BAP aerosol condensed onto carbon black particles and expose time-pregnant dams to the particular carcinogen. And then, of course, for a mouse GD20 or a rat GD21 represents PND0. And we then follow early dysregulation of developmental gene regulation in terms of determining the ontogeny of glutamatergic receptor subunit expression, pre-weaning. And of course, if you note here, it's very, very clear because cortical synapse consolidation is occurring within those first 2 postnatal weeks. And I think you've seen examples of that from several investigators earlier today. And of course, we look late life at behavioral deficit phenotypes, okay? And our mouse model is a CPR mouse. It's just a C57 black mouse where the NADPH associated with the cytochrome P450 system of monooxygenases has been deleted. But I won't show you any of that data today. I'll simply show you wild-type CPR mice, control and expose wild-type CPR mice. And so when you do expose the time-pregnant dam, and you look on postnatal day 3 in whole brain and in the dam in the liver, you do see a concordance with regard to the generation of reactive BAP metabolites. These are the metabolites that we suspect are operable with regard to inducing the neurotoxicity associated with PAHs, and shown here are the percent total metabolites in terms of the composition for the 3-hydroxy, 9-hydroxy, the 7,8-dihydrodiol, which many of you know, the 4,5-dihydrodiol, as well as the 9,10. And so you can see that it's highly likely, and the data suggests that maternal toxicity does contribute to the deposition of these metabolites in CNS structures from the mother to the dam. Although in these structures, we do have expression of the aryl hydrocarbon receptor also, and we've reported that previously. Now, to a learning and memory correlate, of course, LTP is the cellular correlate for learning and memory. And when you look at, for an example, littermates, and this is a study that Deanna Wormley did in my laboratory, she was able to convincingly, reproducibly document the fact that, as you can see here, subsequent to a high frequency stimulation in the open squares, these animals produced and showed a very robust LTP. So those are the open squares. But now, animals that were treated gestationally on those 14 through 17 with BAP shown in the green triangles, or TCDD, which is a halogenated aromatic hydrocarbon, or a mixture of the two, shows significant deficits in offspring plasticity mechanisms, okay? And this is NMDA-dependent LTP. And so this sort of gave us a clue as to the mechanisms that were operable here. You also look at behavior. One of the things that study section wants you to do is demonstrate a behavioral phenotype change immediately. And we use initially the schedule control operant behavior task. It's just a lever pressing task where an animal receives a food pellet reward for performance of a task, which is lever pressing. And when we looked at those litter mates from those separate cohorts, we were able to demonstrate, once again, significant deficits in learning behavior, behavioral learning, from a fixed ratio of 1 up to 20. Now you see this FR of 1 and the FR of 5. The animals are able to do the task, control and expose alike. But when you ask them to do something more difficult, as a fixed ratio of 10 or a fixed ratio of 20, the deficit becomes apparent. And so these animals can't even feed themselves to the point where these deficits are robust. Another behavioral correlate that one would like to use, and that we use now, is the idea of this executive function correlate, which is the two-choice novel object recognition task. And to look at executive function in terms of a quick screen, you want to employ this task, and it measures the exploration of novel environments with regard to a single novel object subsequent to two familiar objects being in the cage. And so when you replace one of the familiar objects with a novel object, then the mouse or rat should show preferential gravitation towards the novel object. And when we did this, of course, you can see that BAP-exposed CPR mice exhibited robust deficits in this type of executive function at a dose of 150 micrograms per kilogram body weight. And we have a little clip that demonstrates this. You can see this is the control animal in the novel paradigm. And you see this is the familiar object, the floater, and the novel object is over in the bottom right-hand corner. And so he's grooming himself. You don't see any problems with gait. He comes up to the familiar object and then moves over to the novel object and spends some time there. This is normal behavior. Now, this is in contrast to a BAP-exposed 8-week animal, and you can see there are some problems perhaps with his gait. He comes up to the familiar object, looks at it, goes back to his corner. Now he's approaching the novel object, and this is a change, this is a shock. And the way that this mouse behaves is characteristic of an ASD phenotype. See, it ran to the corner, didn't want to have anything to do with it. And so these are just a couple of the behavioral techniques that one can use. We also have employed a very useful neuronal circuitry task where we utilize the whisker to barrel field circuit. And this is a task that allows us to assess the effects of toxins on somatosensory development. And here you see that circuit. It shows a rat whisking a walnut. Of course, rats sense their environment by whisking. And this synapse proceeds through the trigeminal nucleus up through the thalamus, and then, of course, commences in the barrel field cortex. And this was highlighted in a recent edition of Neurotoxicology. And so when you stimulate the whisker with a piezoelectric stimulator and actually record evoked activity, one sees the following. Now, this is a deconvoluted post-stimulus time histogram where we have broken it up into 10 millisecond epochs, if you will, data bins. And the first epoch from 1 to 10 milliseconds is AMPA dependent. Responses strictly due to the AMPA receptor fall into this category, early latency neuronal activity. The other epochs are NMDA dependent, and I think you can see that it's very clear that in this early latency component, as well as some of these later NMDA-dependent components, which are NMDA-dependent, we see significant differences and deficits in evoked neuronal activity. When you look at neurons from these animals, ex vivo, cortical neuronal preparations, one is then able to verify what we saw electrophysiologically, and that is that in ex vivo BAP-exposed neurons, we see a reduction in the markers particular for this type of plasticity. That would be AMPA and NMDA, and these are just some preliminary primary cortical neuronal cultures which actually show GLUR1 and NR2B staining and co-localization on the confocal, and it's very clear that there is reduced expression of NR2B and GLUR1 in these animals that have been offspring that have been exposed to BAP gestationally. Now, at about this time, and exposure was during neurogenesis, and of course analysis is during synaptogenesis, and on culture day 7, which is a little early, but still within that window. Now, it was about that time where Pat actually became aware of some of our previous work and sort of put all of this into perspective for us. He let us, he invited us to a lab meeting, and Dan Campbell and I sat down and talked at great length and indicated that some of the previous studies where we discerned an effect of benzopyrene with regard to SP1 modulation was brought out. They saw the same thing with the MET receptor, tyrosine kinase. And so as you see here, MET, and Pat showed this slide earlier, MET signaling augments NMDA currents, it enhances LTP, and it actually contributes to glutamatergic synapse formation. And so Pat actually has shown this, but I simply show it to say that upon addition of hepatocyte growth factor, the natural ligand for MET receptor tyrosine kinase, one can see an induction of NR2B expression as well as GLUR1 expression. And so what we were seeing in our animals highly suggests that MET signaling is being impaired because MET is upstream of all of these H receptor dependent mechanisms. So the conclusion was that the regulation of MET expression in development is critical here. And so when we look then at samples from animals, protein samples looking for MET expression in these CPR offspring, we saw the following. And if I can get, yes. Okay, so these first 4 lanes are the control. The top band is the MET band, and the bottom band is the beta-actin, albeit a bit gritty. But you can see this represents a temporal developmental expression profile, if you will, for postnatal days 0, 5, 10, and 15. Pat showed a similar slide here, and so the ontogeny, or the peak of MET expression, is consistent with the data that he showed early, and down here it's just graphically displayed. Now, so you see a peak of MET expression at about postnatal day 10 in our model. Well, when you expose these animals to BAP, now these are offspring, so the time-pregnant dams have been exposed, okay? When you expose them, you see a dose-dependent downregulation of MET expression across the board, as well as alteration in the temporal developmental expression profile. And so we were very happy to see this, and this obviously is very consistent and supportive with respect to the data that Dan Campbell and I and Pat observed in neuronal cultures, where we simply incubated those neurons with benzopyrene. These are in vivo studies, and we're very happy to follow this up. And so what we think basically with regard to glutamatergic regulation or glutamatergic dysregulation as a result of environmental contaminant exposure, particularly with benzopyrene, is the following. And so you have at the E14 to 17 synapse, these are just neurotransmitters. This is glutamate in blue and green, and perhaps serotonin in red, if you like, coming from a projection from another part of the brain. I won't say which, but it's very clear that these glutamatergic receptors receive input and then stimulate neurogenesis, okay? Well, upon exposure to BAP, as I've shown you, BAP is very lipophilic. It initiates signaling through the aryl hydrocarbon receptor and then through the cytochrome P450 system of monooxygenases to basically generate BAP metabolites. And so we've actually documented this such that the effect in terms of postnatal synaptogenesis is a downregulation, if you will, of MET expression and a downregulation of the respective glutamatergic receptor subunits on the presynaptic cleft in the postsynaptic density. And so this, of course, would result in a downregulation of the pleiotropic receptor MET, which is upstream, with a concomitant decrease or downregulation in NR2B and GLUR1 expression. And we've demonstrated this with mRNA and protein. And of course, the consequence of this is that this leads to postnatal deficits in vivo neuronal activity and behavior. And I'm kind of like Mickey, and I have to stop right here because if I continue, I'll go over my time, and of course Beth Ann will throw me off the stage. But sufficient to say that what we plan to do now is to attempt to induce this behavioral deficit phenotype as a function of gestational day knockdown of MET. And so those studies are underway. We'll be reporting on those at the SFN meeting very soon. And we look forward to those results. And of course, I have a number of collaborators here at Vanderbilt and Meharry, Mickey, Dan, Pat, Fred Gingrich. We certainly appreciate their support and our NIH support. And thank you very much for your time today.